Hello everyone. Today's topic for discussion is apogamy, apospory and parthenogenesis, which are the ways of asexual reproduction. We are familiar with this structure of ovule where this embryo sac it is with antipodal cells, then secondary nucleus, egg cell and the remaining synergids. But when the embryo which is formed from different origin that results into the apomictic reproduction, and these are nothing but the apospory, parthenogenesis and apogamy. Then in apogamy the embryo is developed from the cells of the antipodal cells as well as the synergids, whereas in parthenogenesis embryo is developed from the egg cell without fertilization, and in apospory embryo sac is developed from the cells of the integument or from the nucellus. Then in pteridophytes this apospory and apogamy is with some different steps, but before that the life cycle of or alternation of generation of pteridophytes, where the sporophytic generation in which spores they germinate, and again afterward the mature gametophyte is formed, which is with the male as well as the female reproductive parts. After fertilization there is a formation of zygote and new mature sporophyte is formed. Then what about apospory and apogamy in pteridophytes? Here in apospory, formation of gametophyte. Remember apospory, and here the formation of gametophyte from vegetative cells of the sporophyte, whereas in apogamy formation of a sporophyte from vegetative cells of the gametophyte. Exactly vice versa condition is there. This apospory is observed in 7 families of the Filicales order: Polypodaceae, Osmundaceae, Hymenophyllaceae, Cyathraceae, Cyzizaceae, Disconaceae, then Maraceaceae. And this firstly demonstrated by Drunet in 1884 as a natural phenomenon is Atherium filix femina variety clarissima. Since the cells of sporophyte are diploid, the developed gametophyte is also diploid in apospory. But in apogamy, the sporophyte develops from gametophyte without fertilization, and thus sporophyte have the same level of haploidy as the gametophyte. And this apogamy is coined by Berry in 1878. And this phenomenon was observed by Farlow in 1874 in Teres critica. In Osmunda regalis, it is observed that they are not producing only 2n as well as a 3n gametophyte, but 4n gametophytes and sporophytes as well. In apogamy, the embryos arises as the result of fusion of 2 prothallal cells, and usually these are just back of the apical notch, and this is observed in Lastria pseudomas. In Polypodiaceae it is observed in Pteris, then Dryopteris, Pellia, whereas in family Hymenophyllaceae, Trichomenes it shows apogamy. In Isoetes, the embryo from the base of the megasporophilous cells is developed, and this is vegetative origin of sporophytic tissue in Isoetes. So these are the different examples where apospory is observed and apogamy is observed. These are the 2 figures how this apospory observed in Atherium filix femina variety clarissima, where this reproductive structures which are formed from the vegetative cells of the gametophyte, and here the sporophytic tissue that gives rise to the gametophyte with the help of vegetative cells in Teres critica. Then there are the different types of apospore. In sorrel apospore, the protheli produced on pinnules in place of a sporangia, whereas in apical apospore, protheli develop at the tip. That's why the apical apospore. And it is observed in Polystichum angulare variety Pulcherrimum. Whereas apogamy is again of two types. One is obligate apogamy. It is observed naturally due to the non-functional nature of sex organs. Some examples like Dryopteris, Sedentum and Osmunda are there for obligate apogamy, whereas induced, or we can say facultative apogamy, it is induced. Obviously it is induced in gametophytes having functional sex organs. So adaptation in dry condition where there is no availability of water, and that's why there is a failure of a sexual reproduction, and that is the reason they undergo apogamy. Number of factors are therefore they affect on apospore and apogamy. So in apospore, complete absence of a carbohydrate in the growth medium, or we can say this is the starvation medium, low light intensity, as well as the age of the cell. Many of the times young cells like shoot apex and leaf tips, as well the basal part of a shoot or leaf, they undergo or they leads to apospore. And in apogamy, by providing different media, that is sucrose at 4%, that develops common apogamy. Along with this, far red light intensity, which is followed by blue light intensity, is also results in apogamy. And different growth hormones like 2,4-D, GA3, IAA, amino acids like tryptophan, and as I told you, very low concentration of carbohydrates, that leads to the apogamy. Then the third terminology is a parthenogenesis, where the formation of sporophyte from egg without fertilization. It has been observed that in some leptosporangiate form, this apospore is followed by neither apogamy nor fertilization, but it is by parthenogenesis, and this is observed by Farmer and Digby in 1907. In Selaginella anocardia, the archegonia, they fail to open, and that's why there is no fertilization. And here the embryo is developed parthenogenetically. Whereas in Marsilia drummondi, two types of megaspore mother cell are there. One are with haploid number of chromosome, and the few mother cell which are larger in size, and they are diploid. This diploid gametophyte produced by large spores, and they formed archegonia, but they never opened, and this is the result of formation of egg without fertilization, obviously parthenogenesis. In Selaginella spinulosa as well as the rubricollis, here also the same case, that archegonia failed to open and egg directly develop into the embryo, and finally eventually that gives rise to the sporophyte parthenogenetically. This is also observed in Selaginella intermedia as well as the Selaginella langeri by Hieronymus in 1911. There are the two types of parthenogenesis, in which in haploid parthenogenesis, egg cell that which is obviously unfertilized, that develops into the haploid embryo, whereas in diploid parthenogenesis, diploid embryo develops from haploid cell. So these are some references. This paper is focusing more ideas on the apogamy, apospore and parthenogenesis in pteridophytes. So with this I am going to stop here. This is all about the asexual reproduction ways in the pteridophytes, that is apospore, apogamy and parthenogenesis. Thank you.